Hey everyone, Miss Lopez here. Welcome to Chapter 6, Section 2, Part 1 of Cell Respiration. We are going to break this video down into several parts because there's a lot of information that we are covering for each of your flipped videos. Let's get started. We've already covered Chapter 6, Section 1. Now we're going to go into Chapter 6, Section 2, Carbohydrate Catabolism, which is the breakdown of carbohydrates. And we're going to focus on the breakdown of carbohydrates in the presence of oxygen. The process of cell respiration is the set of metabolic reactions used by cells to harvest energy from food. A lot of energy is released when reduced molecules with many carbon-to-carbon and carbon-to-hydrogen bonds are fully oxidized to CO2. Remember that oxidation is the loss of electrons. The oxidation occurs in a series of small steps, allowing the cell to harvest about 34% of the released energy, which is very useful. You can see here in the process of cell respiration how each of these small reactions have a set amount of activation energy to overcome. And in the process of taking this step by step, you have many opportunities to harvest energy either in the form of ATP or in the reduction of NAD+ down to NADH. If the molecule of glucose combined with oxygen was lost all its energy in the bonds in one large step, the problem is that this one large step would lose a lot of that energy to heat. This is the second law of thermodynamics. So by breaking it down into smaller steps, the cell is able to harvest more energy and not have that giant explosion or net loss of heat that it does if it were in one large step. So just a reminder of what you all know about cell respiration to give us a foundation for where we are going in this process. So you know that cell respiration is glucose, C6H12O6, in the presence of oxygen, producing CO2 and H2O. We're going to break down this process and we're going to look at it in two stages. In stage 1, we're going to look at the breakdown and oxidation of glucose. This is going to occur in 3 stages: glycolysis, the oxidation of pyruvate, and the Krebs cycle. Then, after we talk about the breakdown and oxidation of glucose, we're going to talk about the main energy production phase, which is forming that ATP through a process called chemiosmosis. In this video, we are focusing just on glycolysis. Again, looking at an overview of this process, we are going to look at the stages of glucose breakdown and oxidation, and then the actual production of the majority of ATP. So the three steps are glycolysis, which occurs in the cytoplasm, the oxidation of pyruvate, the citric acid cycle, also known as the Krebs cycle. This is going to occur in the mitochondria. And then the actual production of ATP, which is going to occur in the mitochondria, specifically on that inner membrane. So let's look at phase 1, glycolysis, the breakdown and oxidation of glucose. This slide is an overview. Glycolysis starts with one molecule of glucose. You'll recognize this structure from Unit 1 when we did the macromolecules. Remember that from this oxygen here, we go clockwise numbering the carbons. So here's carbon number 1, number 2, number 3, number 4, number 5, and number 6. You'll also remember that for this particular molecule of glucose, we can differentiate between alpha glucose and beta glucose by looking at the hydroxyl group at carbon number 1 and carbon number 4. Depending on which direction that hydroxyl group is drawn. The process of glycolysis involves 10 reactions, and remember that each of these 10 reactions is controlled by enzymes, or what we say is regulated by enzymes. This process of glycolysis is a good example of a biochemical pathway. It takes place in the cytosol, and it takes place without the use or presence of oxygen, which is O2. The final products of glycolysis include 2 molecules of pyruvate, or also known as pyruvic acid, which is a 3-carbon molecule, 2 molecules of ATP produced by substrate-level phosphorylation, and 2 molecules of NAD+ reduced down to NADH. It is this molecule right here that allows for the glucose to be oxidized. Remember from our previous notes that you can't have oxidation, you can't have something gaining or losing electrons without something else to balance that. So in this case, glucose is oxidized down to pyruvate, and in the process, as we strip away those electrons, we're going to donate those electrons to NADH. Here is a simplified version of glycolysis, starting with the 6-carbon glucose. In our flipped assignment and warm-up, we broke down glycolysis into 2 phases, the energy investment phase and the energy harvesting phase. In our energy investment phase, we are going to input two molecules of ATP to get this reaction started. ATP is hydrolyzed, or that last phosphate group is broken off and then donated to the substrate in this first part of this biochemical pathway. What is left is a phosphorylated molecule called fructose diphosphate. Remember that di means 2, so it's a fructose with 2 phosphate groups attached to it. You're also left with a molecule of ADP, which can be recharged later in the process of cell respiration. In the energy harvesting phase, we are going to take the 2 molecules that are result of the breakdown of fructose diphosphate. We're going to take these 2 3-carbon molecules and we're going to continue on a biochemical pathway to using enzymes to break them down ultimately to the final product, which is called pyruvate or pyruvic acid, a 3-carbon molecule. The pyruvate is shown with the 3 carbons, 1, 2, 3. Because we had started off with a single glucose molecule that gets broken into 2 pieces, at the end of glycolysis, you will have 2 molecules of pyruvate as the product. In the process of forming the pyruvate, you're going to get 2 important steps that happen. Number 1 is the reduction of NAD+ down to NADH, thanks to the substrate being oxidized. Another important step is taking 2 molecules of ADP and phosphorylating them into ATP by harvesting some of the energy that is occurring as a substrate is being converted into a product. We can take off that phosphate group and we can use some of the energy that is released in this breakdown to phosphorylate ADP into ATP. Now, this process, when a substrate is being converted into a product in this biochemical pathway and utilizing the energy that comes off that reaction to phosphorylate ADP into ATP, is called substrate-level phosphorylation, and a total of 4 ATP being produced. The 2 end products of pyruvate are going to be further oxidized to harvest even more energy, but that is the subject of another video. This concludes the end of Chapter 6, Section 2, Part 1. Thanks so much for watching.